Welcome friends. Today we will be going to discuss about the important process of developmental biology or that is called as the oogenesis. Basically it is a process of the formation of the ovum in case of the female in case of the female individual. Okay, so we will be discussing this process in little bit quite detail, and we will start this lecture from the previous topic after the formation of the progenital cells. We have already discussed in the spermatogenesis first lecture that the formation of progenital cells, what was the and how the progenital cells origined and how they reached to the genital reach. Already we have seen here. Let us see how the progenital cell reach to the genital reach, and each genital reach will develop into an ovary, and the cell that is present in the genital reach will give rise to the formation of the ovum. Okay, so let us start or let us begin our this session from here only. So with this figure is divided into the two parts. The first part is when the baby is inside the womb, or it is the embryonic stage and the post embryonic stage, that is prenatal phase and the postnatal phase. Okay, in case of the prenatal phase, the development of the development of the ovum starts here, and after postnatal means the after an individual attain its maturity, then after the second part of the oogenesis just started here. Now if we are saying that, let us to begin about the formation of the oogenesis, about the process oogenesis. We will be starting from the progenital germ cells. Okay, so whenever after the formation of the progenital cells, okay, after the formation of the progenital cells, a mitosis process occurs here. A mitosis process hoga, and this mitosis process is occurred after the progenital cells is Welcome friends, welcome to our hello friends, welcome to our the lecture series, and the today's topic is the oogenesis. In this topic we will be discussing about the formation of the ovum. Okay, before starting this chapter, let us understand the background of this chapter. So in this chapter we will start from the specific cell that is called as the progenital germ cells, and in case in the previous lecture of the spermatogenesis part 1, we have already discussed about the journey of the formation of the progenital cells, and we have also discussed regarding the fate of the progenital cells. So we have the two fate of the progenital cells. The first one was, we have already discussed that the progenital cells will be turned into the sperm, and another fate was the progenital cells will turn into the ovum. Okay, after various types, we have also discussed the various type of the factors which are responsible for the differentiation of the germ cells into the into its successive fate. Okay, so we have if we look out into the picture, we will see that we have just presented the two figure. The first figure, this one is the first figure, and this first figure is regarding is about the individual which is inside the womb. Okay, so it is the prenatal phase, and this one is the postnatal, means after the baby is, after the individual attain its maturity, the rest of the process starts here in this figure. So let us understand this topic in little detail. So we will start our journey from the progenital germ cells. Okay, after the formation of the progenital germ cells and its other parts occurs in after after 2 to 7 within the 2 to 7 month. After the when it is attained at the 7th month, then there are almost 7 millions of the Oogonia is there. Oogonia is there, and after that it goes in atresia phase, and during the birth of a baby, it remains to be another 2 million, and then after only 40,000 is survived, and 40,000 will turn into the mature Graafian follicle, and out of these, only 400 of the mature Graafian follicles turns to be an ovum in the life of the female individual. Okay, now let us begin our study in the embryonic stage. The progenital germ cells, which we have already discussed, under some influence it will turn into be other female fate. Okay, under subsequent mitotic division, the progenital cells under subsequent mitotic multiple mitotic division, it will turn out to be form it will turn out to form the fate of the oogonia. Okay, and this oogonia this oogonia is wrapped in a you can say in the envelope that is called as is wrapped in an envelope, and out of these, just we can see here there are the 4 cells or 5 cell could be here, and out of these, the best possible cell, the best healthy cell will turn out to be at the primary oocyte, and the rest cell will die. Okay, and this out of this 4 or 5 cell, the base cell will turn out to be the primary oocyte. Okay, and after the formation of the primary oocytes, okay, there are certain granulosa cells are there, which are the certain granulosa cells will be there, which will be coming just periphery of the oocytes, primary oocytes, and it will make a layer that is called as the layer of the granulosa cells. And what are those cells? These are the flat epithelial cells. These are the flat epithelial cells, and that is called as the granulosa layer. Okay, now this granulosa layer will just expand, this cell will start to expand, and it is estimated that the volume of this cell, the volume of this cell increase 500 times, and the diameter of this cell increases from 10 to 80 micrometers in size. So this is about the and before we come to the another process, okay, before we come to the another process, okay, let us also discuss what are those factors, because if it is increasing in the number, okay, if it is increasing in the number, what is the reason behind this, how the granulosa cells got accumulated, and why this layering of just getting they are getting so much layers around the oocytes. Okay, so this is another factor that is called as the GDF9, and this GDF9 that is called as the, you can say, the growth differentiation so this is the growth differentiation factor. It is a it is a type of the TGF factor, transforming growth factor, or TGF. It is a it is a member of TGF. Okay, it is a member of TGF, that is called a transforming growth factor, and this is responsible for the accumulation of the granulocytes, or you can say the epithelial in flat epithelial cell around the developing primary oocytes. Okay, and this will keep on increasing in number. This will keep on making the layers in the subsequent stage. Okay, now here, after the transition of the oogonia, after the transition of oogonia in oogonia to the primary oocytes, the cell will no more go into the mitotic phase. The cell will no more go into the mitotic phase, because at this time that we have already discussed in the previous lecture, there was another factor that is called the NRA. There was another factor that is called as an RA, just secreted from, transcribed from the mesonephros, and this is just just get accumulated over there, and this is responsible for the meiotic phase, of the oogonia, the primary oocytes. Okay, so at this stage, at this stage, the primary oocyte will start or will undergo the meiotic division. Okay, so it will undergo, and this will let the RA will let the cell to undergo in the meiotic division. Just remember, in the case of the the spermatogenesis, this RA was being degraded by the cytochrome 26b1. It was being degraded with the help of the cytochrome 26b1, and that was the reason the sperm cell does not go in the meiotic division, the phase of the cell cycle. And but here there is no any formation of the cytochrome 26b1, and that's why, and this is the reason why the cell of the primary oocytes undergo the meiotic first division. Okay, so after the first meiotic, means the cell undergo the first meiotic. Okay, and the first meiotic, in the but whenever the cell was in the cell was in the stage of the prophase. Okay, when the cell was in the stage of the prophase 1. Okay, in the case in the stage leptotin. Okay, so it get arrested over here. Okay, in the leptotin stage, the cell get arrested here. Okay, the cell get arrested. Now, after, what will happen after the cell gets arrested, and what could be the specific factor responsible for the arrestation of the cell? So, in case when we will read about some articles, then it was found that is the follicle stimulating hormone, which is secreted in the embryonic stage, is responsible for arresting of the primary oocyte in the leptotin stage. Okay, so this was the story which we often study in case of the in case of the development of the ovum in the embryonic stage. And after this, we will just go to the individual after it get to the individual after attaining its maturity. Whenever the individual is attained its maturity, then this cell will starts to develop, it will starts to increase, or thus it will starts its further development. Okay, so at this stage the cell cycle is arrested, and in the case of the arrested, the female baby is born, and the cell remains to be in this stage or arrested state till it just got its maturity, or till it got its puberty. Now, after the baby has attained its maturity, so let us come here. What will happen after the baby has got its maturity? Now, And we can also say the cell, the cluster of the cell, that is called as the layer of the granulosa, layer of the primary epithelial flat epithelial cells will call as the, and this will be called as the primary, you call the primordial follicle. This will be called as the primordial follicle. Okay. And after the maturity is attained, the further development of the primordial follicle starts here. But it is to remember here, the cell which is in the stage of the primary oocyte would remain there in its primary state. It will not go, it will not divide further till a signal is obtained. What is that signal? What could be the different layer, what could be the different enzymes or the factor which could initiates the primary oocyte to develop? Okay, so we will discuss it in the next stage. So after this is after the baby matures, the primordial follicle, which we have brought it from the after the birth of a baby, the primordial follicle, that will be called as the cell which is in overlapped with the granulosa layer, that could be that could be called as the primordial follicle. Okay, so the primary after the under the influence of the various hormonal under the influence of various hormones, the primordial follicle will start to grow. The primordial follicles will starts to grow, and this will keep on growing and accumulating under the influence of GDF9, and the layer of the granulosa cells will get maximized and the layer of the granulosa cells will maximize, and this will be called as the growing Graafian follicle. Okay, now what happens thereafter means, in meanwhile there is another cells that is called as mesenchymal cells starts to, there is an scarce and there is an availability of the mesenchymal cells is also there. Okay, the mesenchymal cells will starts to hover around the layer of the granulosa over the granulosa layers, and after the subsequent division, it will make the thin layer of the, you can say, thin layer of the mesenchymal cells, and this is called as the theca layer. Okay, and this theca layer is having the two parts, that is called as the theca externa and the theca in this one is theca interna. Okay, and the cell means the cell which is present over here, that is called as the granulosa cells will differentiate, and they will differentiate and they will starts to divide. After dividing, what happens? After dividing, the cells over here, that is called as the granulosa cells, the cells over here just will starts to divide, that we have already discussed. They will starts to divide, and they will divide in such a manner that the certain gaps and a space will be remaining. And this space, after the subsequent division, this is the space, hollow space. This is the hollow space. Okay, after the subsequent division, the further division, after the further division, the cell over here will accumulate. The cell, the layer of the cell will accumulate, and the layer of the cell will accumulate, and a space, a fluid filled space will be formed, and that will be called as the antrum. Space, the fluid filled space, that will be called as the antrum. Right now till we have got the theca externa, theca interna, okay, and this is the layer of the granulosa cells, and this is called as the cumulus ophorus. That will be called as the cumulus ophorus. Okay, and after, and this will be called as the primary follicles. Here the primary follicles will turns out to be a secondary follicle. So the primary follicle will turn to be a secondary follicle. Okay, and this will turns to be a secondary follicle. Now what happens? Now what happens? The cells, that is the cell that is called as the oocytes. Now this is to remember over here that the cell is still in the case of the primary oocyte. However, the numbers of the changes is being occurred around the cells which is present just around the oocytes, and but the still the oocyte is in the primary oocyte state. Okay, and from here it will starts to divide, means the cell will starts to divide, and initiation of the division of the cell will be performed under the under certain influence. Okay, so before that there is another event which is occurring in the secondary Graafian follicle. Okay, so what is that event? So this is the primary oocyte, and this primary oocyte will starts to transcribe, and the gene which is responsible for the formation of the another layer, that is called as the zona pellucida, and this will the protein of the protein, the glycoprotein, the zona pellucida will, the layer of the protein will be formed around the primary oocyte, and this is called as the zona pellucida. Okay, this is called as zona pellucida, and the zona pellucida, the layer of the zona pellucida is just will thicken from inside to the outside. This is the most important thing. Now let us understand after that what will happen. After the formation of this layer, after the formation of this complex layer, what will happen? The you can say the primary oocytes, the primary oocytes is able to synthesize or able to transcribe a lots of gene, and these genes will be responsible for the you can say the differentiation of the cells, and it also starts to transcribe for the another factor which is responsible for the bringing out the formation of the blood vessels towards it. Okay, so what are those factors which is transcribed, and what are those protein for which the oocytes, the primary oocyte starts to transcribe? So these are, at this stage, primary oocyte synthesizes proteins like leptin, FGF2 beta, VEGF, vasculoendothelial growth factors. Okay, so these are the factors which are synthesized actively with the help of the primary oocytes. Now, up to this stage we have already studied. Now, that the most important thing is after this stage, the cell will starts to divide. The cell will starts to divide means the whatever the cell was in the first meiotic stage arrested in the on the first meiotic state under the influence of LH, luteinizing hormone surge. Under the influence of luteinizing hormone surge, what happens? The luteinizing under the influence of luteinizing hormone, okay, MPK is will be just you can say the MPK, the protein will be phosphorylated and they will get activated. What happens right now? Let us understand here. Under the influence of under the influence of LH surge from pituitary. Under the influence of the LH surge from pituitary, LH will starts to activate MPK, MPF you can say, will starts to activate MPF. What is this MPF? It has MPF has two subunit. The first one is cyclin B and the second one is P34. Cyclin B, P34 is a cyclin dependent protein kinase. Okay, and what will happen under the influence of under the surge of luteinizing hormone which was secreted from the pituitary gland, the MPF will leads to the process of first meiotic division to complete. Okay, and after the cell has just completed its first meiotic division, it will form a two cell, and this type of the cytokinesis of this cell division is of unequal. It will form a two cell. It will form a two cell, that is called as the first cell will have the maximum cytoplasm and another will have the minimum cytoplasm. Okay, so first cell will be called as secondary oocyte. Okay, first will be called as the secondary oocyte. Okay, just it will be called as the secondary oocyte, and another cell will be called as the polar body. Okay, now it will after under the influence of MPK protein. MPK is maturation promoting factor, or you can, it is also called as mitotic promoting factor. So the first meiotic division is completed over here. Now the cell undergo the second meiotic division and it will start to undergo the second meiotic division, but but again before it undergoes the second before it completes the meiotic division, it is again arrested. The cell again arrested. The cell again arrested in second meiotic division. The cell again arrested in the second meiotic division because because of the action of the MPK, the second division had also stopped here, and this will be completed after the ovum will meet with the sperm. So after that, the cell which is in the process of the second meiotic division, and it got arrested over here, and under the surge of, again in the surge of the luteinizing hormone, the membrane of the ovum will degenerate and the mature ovum will come out from the cavity, and this structure will turn to be a structure that is called as the corpus luteum. Okay, this structure is called as the corpus luteum, and it will keep on synthesizing the progesterone, which will be helpful in maintaining the health of the uterus for conceiving the arrival, to welcome the arrival of the ovum, or you can say the fertilized ovum. Okay, so now here the cell, the mature ovum just get released into the fallopian tube, and it will wait for the sperm to meet, the sensual touch of the sperm to meet, and after the sperm penetrates inside the ovum, the sudden, and then what happened, the second meiotic division of the ovum just get complicated, and hence it was the process of the development and the process of the formation of the oogenesis in case of the female individual. Okay, so thanks for watching, and if you like the video, then please don't forget to like, share and subscribe. Thank you.